Good afternoon, everyone. It is great pleasure to introduce one of the finest research from KIST in European Pharma 2015. Here, I'm gonna to introduce my recent research on the neuroprotective effect of a traditional Korean herbal formula. I'd like to begin with the current status and the outlook of plant-based pharmaceuticals. Secondly, I will introduce the natural resources for neurodegenerative disease such as Parkinson's disease. The definition of plant-based pharmaceuticals in Korea is pharmaceuticals from the ingredients and fractions originated from natural products, having new preparation or showing new efficacy, which is regulated by the law of promotion of the research and development of wonder drugs by using Natural Substance Act. Developing plant-based pharmaceuticals in Korea is regulated by the Korea FDA. 7 kinds of plant-based pharmaceuticals were released in Korean market, and their total sales reaches above $150 million US. Among them, Stillen and Jones top the market list with the total sales of more than $100 million US in year 2013. Not only in Korea and other Asian countries, but also in USA and Europe, the plant-based pharmaceuticals are being rapidly clinically tested too. After FDA introduced the botanical drug approval regulation in 2004, more than 200 cases of botanical drugs underwent IND test. Now, let me introduce the natural resources for neurodegenerative disease for Parkinson's disease. Neurodegenerative disease is one of the most highlighted disease group in these days. Many of the diseases in this category are chronic and not fully understood with its basic mechanisms. Among these diseases, I will introduce the Parkinson's disease. Compared to its lack of knowledge and cures, Parkinson's disease is a well-known neurodegenerative disease. This slide showed us the increase of the number of Parkinson's disease, and it will be increased more, according to one of the daily newspapers in Korea. Parkinson's disease is characterized by the neuronal cell death in substantia nigra. It causes lack of dopamine in striatum and impairs the balance of neurotransmitters, resulting in the symptoms of tremor, rigidity, and postural instability. Nowadays, L-DOPA, the precursor of dopamine, is mainly being used to compensate the lacking dopamine in the brain. Along with it, dopamine receptor agonists and anticholinergic drugs are being also used. However, these drugs do not cure the neuronal cell death, but compensate for the lack of dopamine, which is not the cure for basal cause of the disease. Also, these drugs have severe side effects with prolonged treatment, including neuronal side effects and postural impairment symptoms. Therefore, it is desired to develop fundamental treatment for the Parkinson's disease that can protect the dopaminergic neuronal cells from the substantia nigra from damage. Recently, alpha-synuclein is found to be misfolded and aggregated in the brains of PD patients, and it is thought to cause neuronal cell death in PD. Inhibition of such aggregation or inducing its lysis are thought to be the new ways of treatment. UPS and autophagy pathways are recently researched as possible mechanisms that can protect brain from damage in PD. To develop experimental models for PD research, MPTP is a well-established neurotoxicant that affects dopaminergic neuronal system. The main purpose of this research is to find a new drug candidate for PD from natural products. It is not based on simple symptomatic treatment, but to enhance the innate self protection, such as innate protection mechanisms, such as autophagy, so it can protect the neuronal cells from damage in PD. Currently, new drug candidates being developed for neurodegenerative disease, PD as well as AD is found to be one of the majorly focused disease. For experimental design, we used the in vitro and in vivo systems. For in vitro systems, NGF differentiated PC12 or SHSY5 cells were used. Autophagy induction effect and the protection effect of neuronal cell death induced by MPP plus were measured. For in vivo assays, MPTP induced and the 6-OHDA-induced animal model in C57 BL6 mice, ICL mice, and ST rats. After behavioral test, pole test, lateral test, and the rotation test, the brains were taken out for immunohistochemistry and the detection of protein markers related on PD. Prescription of DG consists of three kinds of herbs: Angelica tenuissima, Polygala tenuifolia, and Dimmocarpus longan. DG shows good activity and patented in Korea, Japan, and USA. The final goal of this research is development of plant-based, orally administrable anti-PD drugs through autophagy induction. Autophagy signaling autophagy signaling pathway is involved various kinds of proteins. To complete lysis, misfolded proteins are involved with several major proteins such as LC32 and Bacillin-1 and MPK and mTOR. For screening the autophagy induction of various kinds of herbs, firstly we tested the level of the transition from LC31 to LC32. Yeolda-Han-So-Tang is main Korean traditional prescription. We modified the prescription into the MYH and modify again to DG. Yeolda-Han-So-Tang is very well famous and well-known prescription in Korea, and it is consists of 7 kinds of herbs, and their constituents are very frequently prescribed and with very little side effects. Yeolda-Han-So-Tang showed good activity in the transition level from LC31 to LC32. And MYH contains 3 more herbs than Yeolda-Han-So-Tang. MYH showed better activity than Yeolda-Han-So-Tang. And in the further research to simplify MYH, DG was obtained. DG consists of 3 kinds of herbs. As you can see in this slide, DG showed a very good activity, time and dose dependently. Three kinds of herbs of DG showed the activity respectively, and also they showed the activity synergistically. And then to check the DG data showed false positive or not, we used the 3MA reagent. 3MA reagent is representative of autophagy inhibitor. When co-treatment with 3MA and DG, LC3 disappeared, which shows the autophagy DG is a true autophagy inducer. And then next, we tried to find the best ratio of three herbs. We tried to test the activity with the various kind of ratios. As a result, 1:1:1 ratio of three kinds of herbs show the best activity. And to check, the DG shows the activity with merely the cell proliferation instead of cell protection. So these data showed us the DG showed the cell protection induced by only MPP plus. Next, the animal test is crucial to find out drug candidate, the efficacy. MPTP model is well established Parkinson's disease model. Acute treatment of the MPTP shows the severe symptom of Parkinson's disease. T-turn time have increased in the MPTP treated mice group compared to control, but it is decreased with co-treatment with the DG significantly almost to control. TLA test and load test led us to similar conclusion. After behavior test, the brains were taken out for immunohistochemistry. In IHC data, the recovery of the dopaminergic neuron cells significantly are shown in both the stratum and the SNC. Lopinirole is a dopamine agonist and is used in clinic with L-DOPA. So in our research, we used lopinirole as a positive control. In behavioral tests, DG treatment group showed good activity similar to a lopinirole group. And next, we tried to get the best preparation in terms of efficacy and physical properties, which is 20% ethanol in water extract, named it Winex. On the other hand, the DG is 100% of water extract. In vitro efficacy test, the Winex showed good neuroprotective effect similar to lopinirole. And in vivo test also showed us the good activity and the similar activity with DG. In 6-OHD-induced rat and mouse model. Autophagy signaling pathway involves various kind of molecules, especially for DOR and LC3 gate, backlin, and MPK, and mTOR. For in vitro mechanism studies, we next showed upregulated these kind of proteins and downregulated the mTOR protein. And also in the DG extracted case also showed these kind of markers upregulated and mTOR downregulated, result in DG induced the autophagic flux to complete lysis the misfolded proteins. And in vivo mechanism study also showed us the similar and same result in mice model. In detail, LC3, Becklin-1, and TH. And in immunohistochemistry result, WinX showed us the TH positive cells increased in both SNC and the striatum lesion. In vivo result also showed us a similar result. And finally, we tried to find out the active component of WinX. We used the activity-guided isolation method. From the methylene chloride fractions, the active component was isolated successfully, and we illustrated the chemical structures. In summary, WinX improved the behaviors in various kind of PD animal models or in vitro systems. It is thought to be through neuroprotection and autophagy induction. And also, we tried to carry out the standardization of raw materials. All kinds of the constituents are investigated, and the standardization is finished. And also, we tried the safety test with WinX, including a single dose oral toxicity and the 4-week recovery study and the 13-week repeated dose oral toxicity, and no toxicity was found. IPs and the publications are shown in this slide. Thank you for your attention.